Hey, how we doing everyone? Welcome back to the Bearded Medic. So today we're going to be getting into the adaptive response, which is the more specific response to a pathogen, and it's what follows after the innate response to a pathogen. In this video we're mainly going to be looking at the process of antigen presentation on MHC2 and the activation of T cells in the lymph node. We'll mainly be following the action of the dendritic cell. The reasons will become clear further along in the video. So as we've said, whenever a cell samples itself, it is displayed on the MHC1 molecule. This is for every nucleated cell. We also have the MHC2 molecule, which is expressed only by antigen-presenting cells. These are your macrophages, B cells, and dendritic cells. Antigen presentation is the bridge between the innate and adaptive response. So let's look at how antigen is presented on MHC2. This is done during the process of either phagocytosis or autophagy. Autophagy is the process of cell recycling. So damaged or inefficient organelles are digested and processed in what's called an autophagosome to keep the cell healthy. A pathogen can also be engulfed into an autophagosome during autophagy. It can be because the pathogen has either broken free during phagocytosis or that phagocytosis just hasn't worked properly. When trapped in the autophagosome, just like phagocytosis, a lysosome fuses with the autophagosome. And remember what our lysosome contains: antimicrobial peptides and hydrogen ions to break down the pathogen. When this happens, a vesicle containing MHC2, which has been produced in the endoplasmic reticulum, fuses with the autophagosome, and the digested peptides of the pathogen is sent to the cell surface membrane bound to MHC2 to be presented. The main difference between MHC2 presentation during phagocytosis and autophagy is for phagocytosis, the MHC2 fuses with the phagolysosome. So our antigen presenting cells are displaying pathogen on MHC2, but in particular our dendritic cells are special. They can do what's called cross-presentation. They can process pathogen on both MHC1 and MHC2. The reason why this is useful will become clear later. So equipped with antigen presented on their membranes, these antigen presenting cells need to go where T cells reside, and that's the lymph node. To allow these antigen presenting cells to travel to the lymph node and increase the chances of activating T cells, chemokine receptors and what's called co-stimulatory molecules are upregulated. These molecules are either upregulated due to recognition of foreign pathogen by toll-like receptors, or when our type 1 interferons alpha and beta bind to the cell. The co-stimulatory protein B7, made up of B7.1 and B7.2, are upregulated as a result. Co-stimulatory molecules are needed to activate T cells. The chemokine receptor CCR7 is also upregulated, which allow these cells to travel to the lymph node to activate T cells. So antigen presentation on MHC2 is only done by macrophages, dendritic cells, and B cells, as they're the only cells that express MHC2. This is either due to autophagy or phagocytosis. Recognition of pathogen also leads to upregulation of the B7 co-stimulatory protein and the chemokine receptor CCR7. Dendritic cells are special as they can cross-present pathogenic peptides on both MHC1 and MHC2. So now the cell is ready to go to the lymph node and activate T cells. But what are the T cells doing all this time? After being taught what to recognize in the thymus, they circulate in the lymphatic system. Each T cell has a T cell receptor. Each T cell recognizes a different antigen via this receptor. They enter the lymph node at the high endothelial venules. At this area there are many receptors that leukocytes can bind onto, which makes it more likely that they can enter the lymph node. For example, we have CCR7, which guides our T cells to the lymph node. Remember, this is also expressed in our dendritic cell as well, so both cells can go to the lymph node. So now we have our dendritic cell and T cell in the lymph node. In the paracortical area of the lymph node, our T cell with their T cell receptor interacts with the dendritic cells presenting antigen. If the T cell does not recognize the antigen, they just leave via the efferent lymph vessel and continue recirculating. Eventually, a T cell with a T cell receptor specific for this antigen presented on the dendritic cell will come along. This is called clonal selection, and now this T cell needs to be activated. This next concept is key. The MHC molecule presenting the pathogen will determine what type of naive T cell is activated. MHC2 is only recognized by T cells which express the CD4 receptor. Hence these cells are called naive CD4 positive T cells, or T helper cells. MHC1 is only recognized by T cells which express the CD8 receptor, hence these cells are called naive CD8 positive T cells, or cytotoxic T cells. Just remember the number 8. This is why dendritic cells being able to cross-present antigen is special. They can activate both naive CD8 positive and CD4 positive T cells. So now let's have a look at how each cell is activated. Activating a CD4 positive T cell can be narrowed down into a 3-step signaling process: activation signal, survival signal, and differentiation signal. Activation involves the binding of the T cell receptor to the foreign pathogen presented. The CD4 receptor on the CD4 positive cell recognizes that the antigen is being presented on MHC2. The survival signal is when the co-stimulatory molecules from the dendritic cell binds onto ligands from the T cell. So we already have the B7 proteins from the dendritic cell. They bind onto CD28 on the T cells. This causes more co-stimulatory molecule upregulation. The one for dendritic cells is called CD40, and this binds onto CD40L, or ligand, on the T cell. This signal means the T cell is encouraged to survive. The dendritic cell then releases cytokines onto the T cell. Depending on the cytokine, the CD4 positive cell will develop into a specific T helper subtype. Hence these cytokines that are released are called polarizing cytokines. Each T helper subtype carries out a different action which we'll get into in another video. As the differentiation signal is given, the new T helper cell subtype releases the cytokine IL-2 onto itself. This is to encourage it to replicate and initiate the next phase of the response. But what if you want the CD8 cells? They go through a similar activation process to CD4 cells. Their T cell receptor binds to presented antigen, and their CD8 receptor makes sure this antigen is bound to MHC1. The survival signal is the same with the co-stimulation, but as these cells are particularly destructive, they also need the help of CD4 positive cells releasing IL-2 on them to be activated. Think of it like a form of 2-step verification. You need both the CD4 positive cell and the dendritic cell to activate them. This is because CD8 positive cells are highly destructive and specific. After activation, they can only bind onto cells that express the MHC1 with the exact same antigen on them that led to their activation. When it finds a cell like that, similar to natural killer cells, they puncture the cell using perforin and then release granzymes to trigger apoptosis of the cell. So you can think of cytotoxic CD8 positive T cells as hitmen, selectively killing infected cells only when they've been told what to look out for. One thing to bear in mind when it comes to T cell activation is that we need all 3 signaling steps to happen. The activation signal, the survival signal, and the differentiation signal are all needed. This is to prevent the immune system attacking itself, because the antigen presenting cells can also present self-peptides, and this is why we need the co-stimulatory molecules. Just think foreign antigen equals co-stimulation equals T cell activation and adaptive response. Self-peptide equals no co-stimulation equals no T cell activation, as we don't want the body to attack itself. Most cells that respond to self should have already been destroyed by the time the T cell is developed. But the ones that survive and come in contact with dendritic cells become what's called anergic because there's no co-stimulation. This means they become ineffective at mounting an immune response. Again, this is so the body doesn't attack itself, and it's a mechanism of what's called peripheral tolerance. So that was a lot. Let's review what we've gone through. Antigen is only presented on MHC2 by antigen presenting cells after the phagocytosed a pathogen, or a pathogen is gathered in the process of cell recycling known as autophagy. This cell then goes to the lymph node as certain molecules like CCR7 and the B7 proteins have been upregulated due to pathogen recognition. In the lymph node, a T cell that recognizes the foreign antigen binds. CD4 plus helper cells recognizes foreign antigen on MHC2. So dendritic cells, B cells and macrophages can activate CD4 positive T cells. Dendritic cells are special as they can cross-present antigen on MHC1 for CD8 positive T cells to bind to. Activation of CD4 positive T cells is a 3-step process. activation, survival, and differentiation. For CD8 cells, they bind to MHC1 instead and need IL-2 from CD4 cells to become fully activated. CD8 cells are highly specific and kill via perforin and granzymes. And to prevent autoimmunity, if self-peptide is presented, the T cell becomes anergic due to no co-stimulation. Immunology is quite a big process and can get very confusing to wrap your head around. But if you have any questions about it, be sure to get in contact so they can be answered. Also remember to test yourself over on Instagram with the quizzes. If you made it this far, thank you for watching the video. If you found the video helpful, be sure to drop a like and make it known. Subscribe to help support the making of future videos, and leave some feedback in the comments to make these videos better. Thank you very much for watching, and see you in the next video. Goodbye.